So Entos Pharmaceuticals is a company that I founded in 2016 in Canada. Basically built upon work we did in my academic lab and in collaboration with a Canadian virologist named Roy Duncan, who discovered a fusion protein that's really unique and that we used to basically create a new gene delivery technology called the Fusogenics proteolipid vehicle. And so Entos have been basically building this technology as a platform technology for use in a wide variety of applications, ranging from cancer to rare disease to infectious disease. So in early 2020, Entos was actually working to bring a cancer therapy to the clinic when the pandemic hit. Entos's R&D operations were mainly based at the University of Alberta. So of course, when the lockdowns happened and we were actually being forced to shut down, we basically actually, the university came to us and said, you have to shut down unless you're working on COVID-19. So speaking with the co-founder, Roy Duncan, who's a virologist, we sort of said, we haven't worked on vaccines before. Was there an opportunity to use the Fusogenics PLV platform to develop a COVID vaccine? Everybody got really excited about it. And so we decided, this is before we founded Aegis, to start working on a COVID vaccine. Shortly after that, I met Aegis's co-founder, Hong Jiang, and together we decided to found a spin-out company of Entos called Aegis Life. So this is maybe February, March 2020. And so basically the goal of Aegis, so Aegis owns the exclusive worldwide rights to the Fusogenics PLV technology for all infectious diseases worldwide, except for Canada. And that except for Canada was really important because we had already been talking, Entos had already been talking to the Canadian government about supporting the COVID-19 development program. So we founded Aegis as a spin-out, basically, with exclusive rights to all infectious disease. But our first, obviously, important and exciting task was to develop a differentiated DNA-based COVID-19 vaccine. And of course, in those early days, we knew very little about the virus and what would happen. And so we were moving forward with a lot of excitement and made a lot of progress in a very, very short time. And I'll get more into where the vaccine development went after. So obviously, Hong left the company in early 2023. Currently, Aegis is being run by myself and sort of helped by the Entos management team, because our research and development activities are co-located with Entos at our Science Park Road R&D facility in San Diego, California. We have this, a suite of advisors still, including Roy Duncan, who is the discoverer of the fusion technology that underlies Aegis's technology. So I think, we're really excited still about the potential application of genetic medicines to disease. I mean, obviously during the pandemic, we saw that almost everybody on the planet took a genetic medicine as a part of an RNA vaccine to protect against COVID. And many people, as a part of our vaccine or trials, took a DNA-based genetic medicine for COVID as well. And there are several very exciting applications outside vaccines where genetic medicines can be leveraged. A couple examples are using gene editing to go after residual disease for HIV, or as an example of our work with the Gates Foundation, encoding therapies in DNA or RNA and delivering them so the body makes their own therapies. And so again, there's a large number of potential exciting applications that a genetic medicine platform like Fusogenics PLV could be used for infectious disease. And we're obviously still very excited about the potential to be able to harness this. So if you look at the genetic medicine landscape of what's available, there are really only two major platforms that have been used traditionally for genetic medicines for infectious disease. And these are basically viral approaches. And during the pandemic, we saw adenovirus, for instance, used as the basis for the AstraZeneca vaccine, the Johnson & Johnson vaccines, for instance. And viral delivery does a reasonable job of delivering, DNA to encode antigens that create an immune response. But the huge issue with viruses is that they also create an immune response against the virus. So you can never give another dose that's effective. And so that was the issue with the viral-based vaccines during the pandemic. They really couldn't be boosted effectively because as soon as your body sees the virus the second time, it clears it immediately before it has a chance to make the antigen again. And so that's why the RNA vaccines really gained favor as we moved from a primary immunization standpoint to a booster landscape. And so lipid nanoparticles are basically fat bubbles that are able to deliver RNA effectively, much less efficiently to be able to deliver DNA. And but for RNA, actually LNPs have worked well for vaccines because the way they deliver nucleic acids is using these ionizable lipids that actually potentially a pretty big immune response. And so LNPs were successful in developing the COVID vaccine and were deployed very widely across the globe. PLV offers a completely differentiated way of delivering nucleic acid in a way that has several advantages over LNP. It's basically similar in composition to an LNP. It contains lipids, fats, but where LNP relies on this ionizable lipid to basically break open a membrane to get into a cell, the PLV uses this fusion protein that Roy discovered to really stealthily get an RNA or DNA into cells and can deliver RNA and DNA interchangeably. And with similar cost of goods and scalability as LNP, but then much more flexibility. While LNP is, can be used for delivery of RNA through an intramuscular injection, as I'll show you, LNPs are made with cholesterol, which makes them go to your liver within about an hour of injection. It's almost all in your liver. And this has been a real challenge for LNP in vaccines and many other approaches. It provides some, pretty significant safety issues as well. So PLV provides a completely differentiated platform for infectious disease that allows us to deliver DNA or RNA. And there's a few reasons why we might want to deliver DNA over RNA: cost and duration, et cetera, are two of those. So we see PLV as a really compelling differentiated platform, both for vaccine and non-vaccine infectious disease development. Just a little bit of technical detail on the fusion protein. So I mentioned Roy Duncan. He's a virologist. He works on an East Coast Canadian university, but he worked for years and years on these viruses that don't even infect humans. They infect alligators and birds. And while many viruses that you're familiar with have fusion proteins, like HIV has a fusion protein, influenza has a fusion protein, they use these fusion proteins to get into cells. He discovered basically a completely unique class of viruses. Really, they're called proteinaceous viruses, so they don't need a fusion protein to enter cells. But he found that this class of viruses makes a fusion protein anyway. And because it's completely different from HIV and flu, it's created a fusion protein that's completely different, super tiny, importantly non-immunogenic, which is really important because that allows for redosing, unlike a virus. And most importantly for our uses, when you formulate it into a lipid particle, it allows that lipid particle to fuse directly with target cells, completely avoiding that pathway that viruses and LNPs take through endosomal uptake and escape, which causes all the issues. And so we finally published a huge paper in Cell last September of the whole development of this platform and the use of delivery for DNA and RNA. And we've created basically a platform of formulations that use the Fast protein to be used for a variety of different, cargoes, DNA, RNA, or even protein, and then applications in different routes of administration, for instance, for a vaccine, for intramuscular injection, but other routes of administration as well. I would say, obviously in the vaccine space, LNPs, play an important role. But again, PLV is quite a bit different. And as I alluded to, LNPs use these ionizable lipids basically to get taken up by a cell. The ionizable lipids basically change shape and break open a hole. So they're basically the recipe to make an LNP requires that they're half ionizable lipid. And to make that stable and keep the RNA inside, they need to be about 40% cholesterol. So about 90% of the formulation is locked with an LNP, and that's why these companies, Moderna and everyone else, are fighting over this sort of ratio of formulation for LNP. Because the Fusiongenix LNP uses a fusion protein for delivery, it now doesn't require ionizable lipid. It doesn't require cholesterol. And this does basically two things. It gives us a huge lipid design space, we call, to be able to innovate and create new formulations that are much more safe, much better tolerated. But because they don't contain cholesterol, they don't get actively trafficked to liver. And so, and as I'll show you, when you inject your arm with the PLV, it stays in your muscle. It doesn't go systemically. And the LNP within an hour or so is mostly in your liver and spleen. And so this sort of lipid design space has allowed us to really innovate formulations that allow us to, precision delivery to different organs like muscle, to dramatically improve the safety, which is important, especially for vaccines, to repeat dose, which is obviously critical for either dosing to some sort of therapeutic endpoint or just to boost a vaccine, and to put in large complex cargoes that could be used for, say, this gene editing approach for HIV and others. So I mentioned that because of the formulation of LNP with cholesterol and the fact that the ionizable lipid it actually is a really potent immune stimulator, that when you dose large doses of RNA with LNP, you see some pretty dramatic results. So this, during the pandemic, we saw Moderna's formulation in particular was a very high dose LNP, created some massive immune responses in some people. It's called Moderna arm. And we see in this experiment where we're giving mice, again, this is RNA put into 3 different LNPs. So this is the Moderna one, or this is the Pfizer one, this is the Moderna one. And when we inject them in the muscle, we see that there is some RNA expression in the muscle, but it's all, most of it is in the liver and spleen. And with the PLV, we see expression in the muscle, and we see 0 expression in the liver and spleen. And so the PLV formulations, from a technical standpoint, stay local, exert that activity locally, but have similar potency as the LNP RNA vaccines. And we see that when the LNPs go to the liver, we have a consequence safety. So when we look at cytokines, we see these are dramatically upregulated. And these are not the cytokines that you want during vaccine treatment. These have a negative effect, making you feel sick and can have pretty significant effects. So again, PLV has a completely differentiated, especially in this world now where everybody's being very sensitive about vaccine platforms. PLV offers an alternative that stays, where you inject it and doesn't have a systemic, sort of non-specific response that doesn't help the vaccine activity. And I'll say beyond vaccines, the PLV allows us to basically deliver DNA encoding therapeutic proteins, for instance, in a way where we can have long-term expression. And this is important, for instance, if we want to make, say, an antibody that blocks the transmission of malaria, which is the focus of our collaboration with the Gates Foundation. We've created basically DNA-encoded malaria antibodies that are injected, through an intramuscular injection. The DNA gets into the muscle cells. The muscle cells basically serve as a factory to make this anti-malaria antibody. And we're working with them to increase the potency of these formulations so that the end goal is that, so we know, actually during the pandemic and other places, that if we create antibodies that block, say, COVID or malaria, you can infuse these in a patient. So you can make the antibodies, put them in a bag, and infuse them in the patient, and they work great. But they only work for a short period of time, and they're very, very expensive to manufacture. So the idea here is to create a muscle injectable version, very, very compelling and scalable cost of goods, deployable in low and middle income countries, but would have the same effect because you're encoding a clinically validated anti-malaria antibody. And for other, this thing on the right is basically just a cool experiment we did as part of our Cell paper, where we encoded human follistatin, which actually is a very interesting protein that's produced by your liver after you work out. It basically signals your muscles to get bigger and stronger. We encoded follistatin in DNA, gave mice a single dose, and within 6 months, they became super jacked, really, really strong, doubled their grip strength, basically doubled their muscle bulk. And so many, many potential applications in infectious disease for, say, yearly dosable treatments that can be administered, say, intramuscularly. So Aegis, over the past 5 years, has been mainly working on the clinical development of our COVID-19 vaccine, and I'll get into the details of that, but also working closely with the Gates Foundation on developing a platform around, encoding therapeutic antibodies for the intent of intramuscular injection and long-term expression. And so that's been a great project. We also have one large pharma partnership based on that approach as well. There's also, so obviously during the pandemic, there was a lot of RNA vaccines sold. There's a lot of excitement around, because that's a proven market, to be able to have a differentiated RNA vaccine delivery system. And so we've been developing a version of the PLV that can be used for an RNA vaccine. And we have a large pharma partnership around that as well, and a couple academic collaborations that I'll get into in a sec. So there's obviously an opportunity and a demand for developing newer, better, safer RNA vaccine platforms. And again, we've had pretty good inbound interest on this. And of course, during the pandemic, and the reason we founded Aegis in the first place was to create a DNA-based COVID vaccine that was differentiated from the RNA vaccines because DNA is much cheaper to manufacture. It makes the antigen for longer, so ideally protective after a single dose. But DNA is really stable, so it would be a fridge-stable vaccine. We showed our vaccine was stable in a regular fridge for over a year. And DNA also produces better T cell responses, which should be better in clearing the disease as well. And so we had 3 clinical programs with our COVID-19. The last program was a version called VAX-002 that we tested in a phase 1/2 clinical trial in Canada that we just paused last year due to a lack of funds, and I'll get into that as well. So this is sort of before Aegis shut down operations. This is what we were working on. We had a COVID-19 booster vaccine. Again, as I'll get into, this program was supported mainly by the federal government of Canada to Entos, and so and again, Entos retained the rights to the vaccine in Canada. So we basically received a total of about 52 million Canadian to support the development of the COVID-19 DNA-based vaccine that went to our initial clinical trial in Canada, our phase 2 trial in Africa, and then our coming back with a booster in a phase 1/2 trial in Canada. But as well, we were working with a number of collaborators to develop new programs. So the La Jolla Institute of Immunology is in San Diego. That's where we actually tested our COVID vaccine. A couple of the researchers there being very excited about the technology. And so we worked to co-develop a really interesting pan-dengue-Zika combo vaccine that we have some really nice data on. It's early stage, but a very important project I'll tell you a little bit about. And then we were approached by an academic group that had already developed and commercialized a dog Lyme vaccine. And we're really interested in applying that knowledge to creating basically a human Lyme vaccine based on DNA or RNA. We had some really nice data for that as well. And then for RNA vaccines, we again developed the sort of RNA vaccine capability of the platform, working with a large pharma partner. Of course, Gates Foundation on malaria and HIV blocking antibodies, and then another large pharma partner on the same approach of being able to produce antibodies from an encoded RNA or DNA. So certainly our journey with the DNA-based PLV was a complicated one. I won't take you through the whole history, but we initially went all the way from concept, sort of when we founded, when Entos first started developing it, all the way to clinic. Our first, patient treated clinical trial was 13 months. Pretty much an unprecedented thing for any company of our size to do. The team basically worked day and night for 13 months to make that happen. I think we treated about 23 individuals in that first phase 1. With that very first, so again, put it in perspective, we sort of started the project in February 2020. We locked down a candidate in June after never working on a vaccine before. We saw in the phase 1 trial that it was very safe. We didn't see any substantial evidence of activity, but because of the safety, we and the regulators saw fit to move into phase 2, and the Canadian government, offered support for us to move that into phase 2. We were still in that case in a primary immunization environment. We hadn't yet created a booster. We updated the cargo of the vaccine to be much more potent. So we created a version that was about 100 times more potent than the first one. It was Covagenic's VAX-001B. We initiated a trial in Burkina Faso to test that vaccine. Unfortunately, Burkina Faso was a very, very difficult environment to work in. We had 2 sites there, but the government of Burkina Faso basically had 2 violent coups during the time we were there. Huge instability, huge challenges trying to ship materials both in and out of Burkina Faso. In the end, we had them ship some of the vaccine back after the trial was done, and it was completely basically dead, destroyed. So we unfortunately, the data, we invested a lot of time and effort into that. The data from that trial was not usable. But the Canadian government actually was a huge supporter of the program, really saw potential for it. they're thinking about basically vaccine security in Canada. To be able to manufacture a DNA vaccine in Canada was compelling for them. So they actually supported for us to come back to Canada with a third generation vaccine called VAX-002. This was now protective against Delta and Omicron. And we initiated a phase 1/2 trial sort of last year as a booster. And we immunized, I think, 45 individuals over the summer last year. And again, I guess the other part of it is that during this whole thing, we were forced and funded to be able to develop our own in-house clinical GMP manufacturing. We interact with regulators in Canada and elsewhere. We were able to basically create an optimized GMP manufacturing process that was amenable to regulators, and we're able to manufacture with our own staff and release all of those batches ourselves. And so the data in animals and I'm showing you booster data here, was actually really incredible. So we were able to, with a single dose, protect against lethal challenge, a lethal Delta variant challenge. That was the data that La Jolla Institute of Immunology got really excited about because it contained the humanized receptor for the virus. And so we tried it in non-human primates. We got durable high titers in non-human primates as well. Unfortunately, despite all of our efforts to show that clinically, we were unable to show those same responses clinically. It was too bad because in the phase 1 population we went into in Canada, I mentioned we immunized, I think, 45 individuals at 2 different dose levels. The pre-existing immunity to COVID was just so high. Everybody had been exposed to COVID multiple times, that we weren't able to tease out a good enough response pre and post booster to really substantiate the fact that it was working. And so in the fall of last year, we decided to pause that program and then move on to basically focus on Aegis' other program. So I would say as a part of the whole COVID program, we got substantial support from the Canadian government. I think over 40 million from the Canadian government. Entos invested about 50 million into the program. Aegis agreed basically to pay any of the costs that were outside of the government funding, which adds up to about 16 million. Entos hasn't collected the 16 million from Aegis. It just sits on the balance sheet. And then I'll talk quickly about the last sort of 2 programs we're working on now, and then the current financial situation and what's happening and where I see a potentially to move forward. So the RNA vaccine platform, again, we created the PLV platform actually to be immune silent. And so when we initially used the PLV as an RNA vaccine, it was performing not as well as the Moderna and Pfizer vaccines. But we worked over the past 2 years basically to optimize a version of the PLV that could encapsulate and deliver RNA, so that on the far right-hand side here is the Pfizer basically formulation of the COVID vaccine. And then here is our IM-18 is our optimized version of the PLV delivering the same RNA. And so we were able to achieve basically the same or even slightly better immunogenicity with an RNA vaccine. Most importantly, though, our vaccine stays in the arm where you inject it and doesn't go to the liver. And again, you can see here, these are basically pro-inflammatory cytokines, sort of general inflammation. We can see that the Pfizer vaccine produces a pretty substantial immune response, but the PLV-based RNA vaccine does not. Again, we've had substantial interest from a number of different pharma groups. We have an active collaboration with a large pharma right now, basically to prove this with a couple different new diseases. And I think if everything's positive there, we should be able to license our technology to this large pharma, and the economics for this license will come to Aegis. Quickly talk about the dengue Zika combo vaccine we're working on with La Jolla Institute of Immunology. World-renowned researcher Suzanne Shrestha developed this. It's really important. So obviously, there is a dengue vaccine that's commercialized, Sanofi. It isn't against all the serotypes. And the problem with these related, the dengue serotypes and the Zika virus is that they're very, very similar. And the issue is that if you're immunized against one or you get infected with one, your body produces antibodies against it, But if then you get infected with a different serotype, those antibodies can actually help the virus infect. It's called antibody-dependent enhancement, and it's a horrible thing because there have been lots of examples of vaccines that have been developed. A big example is RSV, where a vaccine was developed given to kids, but then if they get a slightly different serotype of the virus, it actually the vaccination increases the severity of the infection. And so by creating a single vaccine that protects against all the dengue serotypes and Zika, which is a similar virus, you can prevent this enhancement of other infections and protect against all of these serotypes. So Sujin created basically a version of this that's very protective in her work. We basically put her package in the PLV and gave it to mice. And so we saw very, very nice responses. In fact, when she saw the first data, we thought it was a sort of a phase, unoptimized version. She said you could take this version to clinic. It's better than anything else out there. So no other platform was able to achieve that. And again, if we can get Aegis on its feet, then there's a lot of excitement from both from La Jolla Institute of Immunology and us to continue to develop this and partner this with pharma. Lyme disease is the other project we were working on. So we were approached by Richard Marconi at Virginia Commonwealth University a couple of years ago. He again, has been working with Lyme disease his whole career. Developed a commercially viable, commercialized vaccine for dogs for Lyme, but that's protein-based. Was really interested in developing a genetic medicine version of a humanized vaccine using similar approaches that he used for the dog vaccine. We did some initial studies. So he's already shown that this approach works well in monkeys. We encoded it in DNA and RNA and did some initial mouse studies and saw a really nice response. So again, this is a viable program from a well-established academic, and obviously in a market that's very compelling as well. Finish off with our Gates Foundation relationship, and this sort of leads me into the funding. So Gates Foundation approached us 2.5 years ago. Really wanted to, so they developed a number of different antibodies that were therapeutic, including one for malaria. So basically, the idea here is that people around the world get infected with malaria. Everybody produces an antibody response. Most of those antibodies don't do anything effective. Every once in a while, somebody produces an antibody that effectively blocks malaria. And so they isolated this antibody sequence, and they manufactured the antibody separately, gave it to patients in phase 1 trials, and showed that by infusing this antibody IV, you can block malaria transmission. But of course, that is a very expensive way to block malaria in low and middle income countries. So the idea, they got excited about using our technology to code that antibody in DNA, deliver it in an intramuscular injection, and have a very sort of compelling cost of goods to distribute this in low and middle income countries, providing long-term durable protection against malaria transmission. And so they initially gave Aegis a grant for $750,000 in 2023 to sort of look at the feasibility of developing this technology for malaria. The data looked really good. So based on that, they decided to make an investment in Aegis. It was a $5 million convertible note, but it was tranched into in different tranches. The first tranche was $1 million, which we got at the beginning of 2024 to move the program forward. And we moved this program forward to the point where we were getting ready to enter non-human primate studies. I mean, I guess the challenge was the tranche, the trigger to get the remaining 3.45 million out of the investment would be triggered on an investment of 20 million or more into Aegis. And we entered basically the scenario where the market was very, very difficult to raise capital from. And so we were unable to basically get an investment commitment for 20 million. That said, I was able to, based on the data that we got, I was able to actually convince the Gates Foundation to release the second tranche. The problem is that at the same time, we were basically Hongjiang served us with a lawsuit. We disclosed that lawsuit to the Gates Foundation, and they had actually already committed to fund and had set the wire date into Aegis, and they said based on the lawsuit, it's just we just don't want to. This is too much risk for both the company and us. Not necessarily based on the merits of the lawsuit, and I'm happy to discuss that as well. But decided to put the investment on hold. So the convertible note is still in place. but they decided not to release the second tranche until we resolve this lawsuit. But I would say their Gates Foundation still remains, pretty interested in the technology, and so just looking for ways to continue to work with us and their other collaborators to move this program forward. So I'll, the rest I'll just talk to. As far as timing, Aegis was, getting low on funds at the beginning of 2025. And so in January and February 2025 is when I started to engage the Gates Foundation to release this next tranche. We talked about releasing sort of parts of it. And in the end, so we went back and forth with diligence, developing the terms. They finally agreed in June of 2025 to fund the first tranche or the second tranche, the entire 3.45 million. We had all the paperwork finalized. We had the wire date of June 21st or something. And as a part of the disclosures, we disclosed the lawsuit. And we went back and forth, but in the end, they just decided to put it on hold. Unfortunately, we were in a position where I was expecting that money to come in. We kept our team, going. I think we had 5 employees on the scientific team working on this Gates Foundation project. And so we were basically, when Gates dropped out, that put us in a very, very difficult position. We had to let all the staff go. And unfortunately, Aegis was in a position where we had about $1.5 million in current debt. A lot of that to lawyers, some of that in lease, some of that to Entos, and not including the vaccine development costs, just for shared lease space, et cetera. And so, and so we've basically since then, that's when I sent out the email to shareholders saying that we were in this difficult situation. We sort of had these liabilities, and as you rightly said, so in the email I basically said we're going to look at options for either winding down or basically selling our assets or finding a way to keep going. We've been actively engaging potential investors in that time. Obviously, we're still in a very difficult environment for seed and Series A stage investors in the market. And of course, Aegis is not in the most compelling position for investment, even though we have sort of this pending Gates investment. And as one of you mentioned, Michael, we have so Entos owns roughly 6.4%, or sorry, Aegis owns roughly 6.4% of Entos. And so when Entos basically did a licensing deal with Eli Lilly, a convertible note from Aegis into Entos converted into stocks at 6.4% of the company. And so I've been looking for potential buyers and in a secondary sale for that stock. And so I've been able to keep our creditors sort of at bay. I would say our most aggressive creditor is our landlord. And so we've been working with them basically to delay payment until we can sort of find a solution to prevent us being forced into bankruptcy. I do have some, I would say, encouraging news. I sort of approached a number of our current investors to potentially buy the Entos stock owned by Aegis. I did find someone who was willing to buy it. And so the first tranche of that sale closes in a little while, and so that will essentially recapitalize Aegis, allow us to basically satisfy our debt with money left over to potentially continue operations. Now, I think we're going to need to think carefully about what that looks like. I think the biggest risk to Aegis right now, obviously, is this lawsuit, which remains pending. I can give you a little bit of detail on that as well. So basically 2 days before Christmas last year, in 2024, we were served with a lawsuit from Hong. Hong was suing Entos and me personally. Aegis was also a nominal defendant as well. And we working through that. I can't go into all the details because it was I think 114 pages of claims. Most of it, in my mind, was just a tirade around, how she was treated. But I would say there are some significant. Basically, the claims were broadly that me and Entos took advantage of Aegis and Aegis shareholders to bring benefit to myself and Entos over Aegis. And I think if you look at the facts of the situation, all the contracts we had in place, and the fact that both Entos and Aegis were stood up by separate legal teams, I think it's very clear that the situation is probably the opposite. Entos has been sort of helping Aegis out, but that we'll have to work through the legal process to get to a conclusion there. And from what I'm told, that process could take a long time. So it's come at considerable financial cost to defend that, and so that's a part of the issue. But I think, and we're in the discovery phase still. So basically getting millions and millions of documents together to give to the opposing counsel. So we think if it goes out to court, this could take 2 to 3 years. We're hoping to find some sort of reasonable settlement in the meantime, and allow Aegis to proceed. One of the other scenarios that we talked about, and we talked about this sort of shortly before Hong left Aegis, is that we could potentially look at combining Entos and Aegis sort of back together. And in the end, Hong and I couldn't agree on anything, but couldn't agree on how that process might take place. But I think we're still open to that as well. I think, speaking personally, I'm at the beginning of the pandemic, when we went out and we raised money from you, very, very excited about the application of the Fusion Genetics platform in infectious disease. I still remain excited about it. I think, unfortunately, we weren't able to show for the COVID vaccine a clinical benefit, but I think the potential applications in many other areas of infectious disease are considerable. I think this is evident because we have pharma companies sort of interested in collaborating with us to look at the potential here. So I think there's still a good potential to deliver shareholder value back to shareholders with Aegis. It's going to be a bit of a long road, but certainly see that. But I'd also be open to combining the two companies and obviously making things less complicated as well. So I'm so happy to. So that's all I have to say. But I'm happy to. If you guys have any questions or want to discuss any other things I talked about, happy to chat about it.
I'll just say, investors, if you have questions, now's the time to ask them. There's a question mark box at the bottom of your screen which says Q&A. So please share your questions there, and John is here. He can address those right away. John, one question I have is, who is on the board of Aegis? Who is, and how does that look versus the board of Entos?
So the board of Aegis originally was just Hong and I, and when Hong left, it went just to me. So an option. So we've sort of put everything in stasis in the meantime, but certainly open to rebuilding out the board and all that. I think inviting people to join the board at this point,
yes,
again, would be challenging, right? So, but certainly open to different scenarios where Aegis can be successful. And currently the board, Entos is really 3 companies: Entos Canada, Entos UK, and Entos US, and there's 3 different boards.
I see. Well, I think at minimum we must meet when you are here. I'd be interested in offering whatever help I can and maybe brainstorming options. I don't know what we can do, frankly, here, or how we can be of help. But if we can be of help, I'd like to do that. We do have one question. Where is Hong now, and what is her claim for damages?
Yeah, so Hong, I believe, lives in Puerto Rico. So she left in, I think, March 2023. Interestingly, she started a new company that sounds very similar to Aegis, that delivers DNA using some sort of delivery system. And I haven't talked to her at all since March 2023. There is no specific claim in the lawsuit, so we're yet to sort of figure out what she wants.
Really? So she's expecting a jury would eventually determine how much to give. Is that what she's thinking?
I think in this particular court, I think that it's just judge only, so the judge would figure it out, or maybe at some point after discovery they would decide. It seems to me, and this is just my personal thought, is she's just trying to dig for things that she can find. But again, I think we're very confident. We've created agreements that define the relationship between the 2 companies. They all have separate legal representation, and we've been more than fair as we've gone along. Obviously, the fact that I'm on both sides is the issue she's trying to leverage, and an issue I'd be happy to solve.
Okay, so it is things are in limbo right now. Yes, and we won't have resolution for a bit.
So I think as far as Aegis expenses going forward, we're going to basically pay off our debts, get the creditors away, and then and not incur any new costs until we're sure of the business plan going forward.
Right. There's another follow-up from Dennis. What is the possibility of countersuing her? People often get more reasonable if they see a possible downside.
So that was initially my thought as well. I think we're looking into that potential as well. But all of this costs money, and even defending this lawsuit has been a pretty significant financial burden already. There's we delivered millions and millions of documents, so we're looking and we're even from the Entos perspective, we're all of these any company in biotech now is in an extremely challenging environment for raising capital. So I think from all we're trying to preserve capital, I guess is the point, but I 100% agree. So I can't disclose at the moment. I think if you email me, I can potentially share some more details. It isn't closed yet, so this is still in the works.
This is to Jade's question. Michael says no question. Thanks for the detailed information. Well, it's a difficult situation. Yeah.
I think like everyone else, we're I think we have a potential solution to the near-term cash flow and debt issue. We're hoping that the biotech market will start to pick up again. And I guess the other part of it is that the two large pharma collaborations, Entos is doing that work now. And so they're still proceeding forward and could end up in license discussions in the next, say, 3 to 6 months.
What would I mean, you can see the question, but for other people, what would an Ontos Aegis merger look like for Aegis note holders and shareholders? Do we all just become Ontos shareholders at some TBD valuation?
I think the way we initially discussed it in 2023 is that we'd have a sort of an independent investment bank do a valuation of both companies, and then do a merger where Aegis investors would get Entos stock.
So I know for everyone, this is, well, here's another one. I think they're coming thick and fast now. I think you should just go ahead and answer them, John. What is the value of Aegis equity in Ontos?
So the value of Aegis equity, so it's private stock. So there isn't really, Entos doesn't really have a value. So it's whatever anybody's willing to pay for it. But again, because Entos has never done an outside funding round, right? So we don't have any VC investors.
I see. Well, that was smart. Okay. All right. It certainly, so there's another question. Certainly seems to me that there may be a potential claim for a breach of Hong's fiduciary duties as an officer and director of Aegis.
Absolutely. That is a reasonable thought.
Yeah, because we did try to just of course you may be aware, we tried to contact her, no response multiple times. So I'll have to look up those emails. Yeah.
I'd say to any of the investors, if you want to reach out to me directly and chat about this, I'm all ears. I think we're looking for any opportunity we have to resolve that, because I think that is the biggest impediment to sort of moving Aegis forward.
Right. Wasn't the Aegis safe triggered by an outside investment? What was the valuation of that outside investment? No, that's not true, but please answer.
I mean, the value cap on Aegis with Gates was 200 million, which I think was some brilliant negotiating on our part. But the threshold to trigger the second tranche was $20 million investment, I believe.
Okay.
But I convinced them to waive that, so we were good to go.
Yeah. So just so I share my thinking on this, I don't know if you're asking Jade about, or Dennis, I know. I don't know if you're asking about our safe, the safe that we invested in. I don't think that was triggered by an outside investment. But yes,
Oh, sorry. Yeah. So that would be triggered by, it would be triggered by an equity investment. Yes.
yes. So
Or
if
it'd be
we,
triggered by or a sale.
yeah. Oh, he's saying the Eli Lilly investment that triggered Aegis's safe conversion.
Yeah. So the Eli Lilly investment, again, we negotiated with Eli Lilly. We did I think it was 205 million post money on that, making, so that was a very high valuation. Entos has gone to the market now to raise more money, and there's no way we're going to get any valuation, anything like that now. So again, it's hard for me to say on a secondary sale how much Entos shares are worth. We haven't sold any recently, so it'd be great if we were at that valuation. Everything would be great.
Yeah, because we do have Aegis on Carta. The second, we had two investments, so the second part, yeah. Right? Do we? I don't know. Maybe
I mean, I think the best case scenario is that we ride out sort of this downturn and we mature these pharma partnerships to the point of licensing, and then all the valuations sort of come back up to what they should be. I'm sure there was a bubble during the pandemic, and I think valuations were probably higher than they should be, but somewhere in the middle, I think, makes sense.
Okay.
I have a friend who basically failed. He's a gene editing company. They were valued at, I think, 120 million during the pandemic. They just failed raising on a $1 million pre-money.
1 million?
So it's really rough out there.
It is rough.
So we're trying to maintain as long as we can with non-dilutive funding.
I think that's a good idea. And is Canada continuing to fund you?
So their COVID specific funding was done about 2 years ago. Canada is funding other things that we're working on, and so that's certainly helping. We did Entos announced a contribution from the federal government for 66 million, and that sounds like a wonderful number. But the issue is it's reimbursement at 30%. So we need to spend $1 to get 30 cents. So again, in a world where we have capital to spend, it'll be great. But I think as far as short-term cash flow, we're looking for other opportunities.
Okay. So I'll just make one last call for questions. We're almost at the top of the hour. And then, of course, if you have questions, send it to us by email and we'll just consolidate stuff and get them answered. But John, thank you. I think this is informative. As Jade also says, it has been informative. Yeah, one last call for questions, investors. If there's any, ask them now.
Well, let me apologize to you for not giving you more frequent updates. So I'll endeavor to get these updates out much more frequently.
Yeah, no, I understand. Sometimes I'm guilty of that too. So it's okay. I get it. There is a smaller team than is able to do all the work. So let's wrap up. Thank you very much, John, and thank you, investors, for attending. We do have a recording. We can share it on demand to whoever needs it. But thank you very much. Bye-bye.
Thanks so much.
Happy New Year.
Take care, everybody.
I hope this resolves in this new year. So
yeah, me too. Take care, everybody.